Okay, so malaria. You'll have to investigate, and this is in the uh, syllabus. So you'll have to investigate the transmission of a disease during an epidemic. So there are a couple to choose from, including including like Ebola is another one, but you can sort of choose any, but I've chosen to go through malaria today because it's a really popular one that's really interesting to, I guess, research as well. So malaria is caused by a protozoa, and specifically it is caused by the Plasmodium protozoa. The Plasmodium protozoa is found in the gut of the Anopheles female mosquitoes, and it transmits via vector transmission. So it transmits when an infected female Anopheles mosquito goes ahead and bites a human, and from there, so once they've bitten a human, you've got a mosquito bite. The protozoa is the pathogen is transferred into your body. It goes through to your lungs, and the next time another mosquito bites you, let's say another female mosquito that does not have the protozoa bites you, they can take away that protozoa. Like it circulates to them, and that is how the disease spreads. So we've got established like a little summary. So let's have a look at what actually happens. So firstly, we've got the Plasmodium sex cells, which reproduce in the Anopheles mosquito's stomach, forming zygotes in the cysts of the stomach wall. Then these cysts burst and sporozoites travel to the salivary glands of the mosquito, and from there it is transferred to humans when they bite humans. Sporozoites then travel to the liver, sorry, not the lungs, the liver, and they enter the red blood cells and continually multiply. And they can actually cause the red blood cells to rupture as well, because they're continually multiplying within the red blood cells. And infected cells burst, which causes malarial fever. And finally, the cycle continues when the human host is bitten by a mosquito, which passes those mature Plasmodium cells back to to the mosquito. Because what happens is once you have been infected with malaria, the Plasmodium protozoa, it stays within your body, so you can't get rid of it, which is why people see like relapses of malaria, because it stays, it's hard to get rid of, but there have been treatments for it, but now the Plasmodium protozoa has become quite what's the word? It has actually become quite resistant. It has become quite resistant to to antibiotics. No, it has become quite resistant to the treatment, and so as a result, that treatment is people still use it, but it actually is not as effective as when it was first delivered. So you can see in the diagram over there, we've got let me get a pen. I really love using a pen. Hopefully you can see my screen okay. So we've got, okay, here we go. So we've got the transmission to the host that happens here, and then those sporozoites, they go straight to the hepatocytes, to our red blood cells, and they affect our red blood cells in the liver. From there, they do mitotic replication. So they are replicating ridiculously fast in the red blood cells, and as a result, our liver cells rupture, and those myriazytes, which are now mature, they are released. Once they're released, what happens is they undergo sexual cycle to produce gametes instead, and you can see here, so they're producing gametes within the human body. So you've got the ring, the trophocyte, Schizocytes producing the gametes over here, and then it ruptures again, lets it out, and then from there, once a mosquito bites that human, it goes into the mosquitoes, and the gametes mates, they undergo meiosis, they migrate through the midgut wall, forms oocytes, and oocysts, and the sporozoites develop and the cycle starts again. So with all of this, you can clearly see the way in which it continues to sort of circulate, and it is hard to get rid of. It is a disease that it's hard to eradicate, because firstly we've got vector transmission by mosquitoes, and then even after you've got the disease, it remains in your body, and that is why people get relapses, malarial fevers, because it gets not reactivated, but essentially, yes, it comes back up, and as a result the cells start multiplying again, and then they bust and they cause malarial fever and so on. And we will talk a bit about what happens when we look at prevention and stuff later on in the dot point. But you don't need to know like in that much detail. You kind of just want to know what I've got here in the dot points for us, all the sort of the terms here. That's just me going into a bit extra detail, because I personally find it very interesting, and I think it's interesting to sort of understand what's happening at the root levels, but you just have to know what the main points are and how it's transmitted. Just because you can get questions which are asking you, how does a named disease transmit and affect people? Obviously it's going to be worded different, but something like that, and that's when you want to be able to name a disease and go through the steps of how it infects everyone. So now we look at an experiment that you will do in class, which aims to investigate the presence of microbes in different water sources. So basically this is just to sort of give you a summary of what it might look like. Obviously the results will differ based on the areas that you test and your sort of experimental design, but basically when it comes to experiments, you want to have clear and concise aims and hypotheses, right? So aim, what are we investigating? Then hypothesis, what will happen? What will we see? Then your descriptive methods you can write out in an exam. So appropriate detail, they can ask you to write out an experiment in the exam. I'm pretty sure they did that in 2021, if I'm not wrong. So something very similar to this. You then want to look at what your expected results are. Obviously, after you've done your experiment, you would know what your results and you want to remember them. You obviously also want to talk about validity and reliability. So validity and reliability. Validity is the experiment testing the aim, and reliability is, can the results be seen as consistent. Accuracy, to what degree are the results precise, and you need to be able to apply these principles to the experiment. Finally, the variables which you need to understand. So having a good understanding of the variables is important. You should be able to tell them apart. Like especially, I think one thing that always confuses everyone is the difference between controlled variables and having a control. So an independent variable is the one that is being manipulated, while the dependent variable is the one that is being tested. So in this case, our independent variable is the water source that we are using. Our dependent variable is the microbial growth that we want to see in the type of water, so the amount and also the species. We're then looking at the controlled variables, so things that we're keeping the same. So in each sample, we have, for example, the same amount of water, the same temperature at which we are culturing those agar plates, and also doing it for the same time. Finally, controls. We can have a control can be a sealed, unopened agar plate which has nothing growing on it. That can be a control, because ultimately that will tell you that there's no bacteria grown on it because it's been unopened, it's sealed, in comparison to the agar plates that have got the sterilized water, pond water and seawater. So this might differ based on what your school decides to use, like if they just want to use, like, I don't know, water from a nearby lake instead of a sea, whatever works. But you kind of, you'll probably have these 4 sets where, oh sorry, you'll have these 4 sets essentially.